Good morning. Good morning, everyone.
Morning.
Good to see you. I hope everybody's doing well. Looks like we're going to have another beautiful afternoon today. Yes, if you could type your name or your nickname into chat for me, that would be great. Thank you. Very good. Okay. So today we're going to be finishing up the lab exercise that we started last time, which was about the way we use ultraviolet radiation to control the growth of microbes. Before we jump into that, though, let me ask if anybody has any questions about any of the material that you have been viewing on the playlist, or any questions about quiz material or anything like that. Feel free to type it in the chat if you do, and we can talk about it. I'm going to pull up our Blackboard page so we can take a look together at our upcoming schedule, at the week we have ahead of us. So right now on your screen, you should be seeing our course content page on Blackboard. I'm just going to scroll down. Remember, there's a content folder here about your pathogen assignment. Don't forget that's there. We're in the week of March 8th right now, and I'm going to go ahead and click on next week's folder so we can take a look. Remember, next week we have a really busy week, an unusually busy week. On Monday, we're going to be talking about controlling microbial growth, specifically with chemicals, with chemical disinfectants. But on Wednesday and Friday, you have exams. So on Wednesday, you'll be taking the third lecture exam. And on Friday, you'll be taking the second lab practical exam. So 2 exams scheduled for next week. That means there's no lab homework next week, just the 1 lab meeting on Monday. And no lab question assignment associated with that. Here's the links for the lecture exam and the lab practical as usual. So there's no quizzes to take next week, just those 2 exams, no quizzes and no lab homework questions. But we will meet on Monday for lab to talk about disinfectant chemicals. Okay. All right. I don't see any questions, so I'm going to go ahead and move into our lab slides. If you remember the last time that we were together, we talked about this physical method of ultraviolet radiation, and we talked about where ultraviolet radiation falls on the scale, the spectrum of different types of electromagnetic radiation, all the way from gamma rays to AM radio waves. And we said that ultraviolet radiation, while it's invisible to our eyes, you can't see it, we know that it's coming from our sun. And we know that it belongs to the types of electromagnetic radiation that are capable of penetrating living tissue. Now, ultraviolet is the weakest of the types of electromagnetic radiation that can penetrate tissue. It's only capable of penetrating to a depth of about 2 millimeters. So it's not like X-rays, it's not like gamma rays, it's not an ionizing type of radiation that just goes right through the human body, for example. It's a type of electromagnetic radiation that can penetrate only to 2 millimeters of depth. So for us as humans, the cells that are vulnerable to ultraviolet radiation from the sun are the cells on the outside of our skin and the cells in the outer layers of our cornea. Very different for microbes though, right? Microbes are much, much smaller than we are, obviously. Only a couple of micrometers in diameter. And of course, a micrometer is only 1/1000th of a millimeter. So ultraviolet can penetrate to 2 millimeters. So these cells, these microbial cells that we're talking about, the ultraviolet radiation can pass right through them. And it's going to have one of two effects. First of all, it's going to damage their DNA. That's what we finished up talking about last time. It's going to damage their DNA. And the question is, how badly damaged will it be? Because if the DNA is damaged beyond repair, the cell will die. So ultraviolet can damage microbial cells and it can kill microbial cells. If the DNA damage is not too bad, though, if it's not too severe, there are repair mechanisms inside of microbial cells, repair mechanisms that can go through the DNA molecule and repair the damage and restore that cell to health, essentially. So UV radiation is a good way to disinfect surfaces, to kill microbes, but it only works on smooth, hard surfaces because it has this one big drawback, which is poor penetration. It can only penetrate to a depth of about 2 millimeters. Cells at this within this depth will have their DNA damaged. So again, in us, that's skin and our eyes. Remember though, you and I do have a protective mechanism in our skin cells, don't we? We have some protection from ultraviolet radiation in the form of melanin. Remember from anatomy and physiology that the epithelial cells in your skin have melanocytes sort of scattered throughout, and the melanin that gets produced will absorb ultraviolet radiation, so it cannot go on and damage DNA. Melanin is going to be produced at a higher rate the more you're exposed to sunlight and ultraviolet radiation, which is why you tan or why your skin darkens when you get into ultraviolet light, ultraviolet radiation. But tanning is only protective to a certain degree. Tanning is only going to protect you to a certain degree. And when you spend a lot of time in ultraviolet radiation, your DNA in your skin cells is getting damaged. And of course, that can lead to skin cancers later on, which is why we're taught now, if we're going to be out in the sun, to put a protective sunscreen over our skin to prevent the ultraviolet radiation from penetrating. Our microbial cells, though, our neighbor microbial cells, they don't have that protection. They don't make melanin. They can't use sunscreen. So they're very vulnerable to ultraviolet, and we can take advantage of that by using ultraviolet in our environments that we live in, certain environments anyway, in order to disinfect these smooth, hard surfaces. All right. So let's talk about what exactly is happening to the DNA molecule when it gets hit with ultraviolet radiation. Just as a quick review of the structure of DNA, remember DNA is a double-stranded molecule in living cells, and living cells all contain DNA. this DNA molecule, this double helix, is held together through what we call base pairing. So on each strand of the DNA molecule, there are what we call bases jutting off of it. And you can see in this diagram, the bases have been color coded: red, blue, yellow, and green. There are 4 bases in DNA, what we call T, A, G, and C. That stands for thymine, adenine, guanine, and cytosine. The bases pair with each other, not one to one. It's not that green pairs with green or red with red, but instead they pair in a complementary fashion. So thymine pairs with adenine, that's T pairing with A, and guanine pairs with cytosine, G with C. Now, every so often in the strand, this double-stranded molecule of DNA, there will be an identical base, like this yellow one and this green one, that repeats. Remember, the sequence of the bases in the DNA molecule is very random, very jumbled to create our genes. So every once in a while, you'll see 2 thymine bases specifically that are side by side in the molecule. You could even have 4 or 5 of them in a row. It's the thymine base specifically that matters when we're talking about ultraviolet radiation. So in this diagram, it's the yellow bar. You could see 2 thymines here side by side, and they are base paired with the green adenines. That's perfectly normal base pairing. Now, when the ultraviolet radiation, when those rays hit the DNA molecule, anywhere that 2 thymines are sitting side by side, this ultraviolet light is going to cause them to separate away from the adenines and attach to each other. So this happens. Essentially, the double strand is broken, and the 2 thymines are going to bind to each other. This is what we call a dimer, and it's specifically a thymine dimer. And this is what ultraviolet radiation does to DNA. There's a second image down here which just shows 2 thymines drawn out side by side. Again, note that they're jutting out perpendicular to this pink strand, which is the backbone of one of the strands of DNA. So both of these thymines are on the same strand, and they're sitting side by side. When the ultraviolet rays come in and hit that DNA molecule, anywhere where there are 2 thymines side by side, you're going to get this. They're going to bind not to the opposite strand, not to the adenines in the opposite strand, but they're going to bind to each other. That's what we call a dimer, and that is specifically a thymine dimer. Now, why is this a problem? Well, obviously, there's a problem right in the location where the dimer is. But there's another potential problem when that cell prepares to replicate. Whenever a cell is getting ready to divide and create new cells, the DNA molecule has to be copied. It has to be replicated. That's so each of the new cells gets their own copy of the DNA molecule. And there's an enzyme that does this work. It travels along each of the strands of existing DNA, and it creates a new strand of DNA. So you end up with 2 DNA molecules at the end. Take a look at this image right here. This green structure right here that looks like a ladder, this represents the double helix of DNA, 2 strands with bases sticking off that are complementary binding with each other. A with T, G with C. The first thing that happens when DNA is copied or replicated is those 2 strands are separated. And then an enzyme comes along and it goes down each strand and it pairs up the base that is complementary to what's on that old strand. So wherever there's an A on the old strand, the enzyme will put a T. Wherever there's a G on the old strand, the enzyme will place a C. And pretty soon you have a brand new strand built on the old strand. So we start with one double helix, and by the end of this process, we have two molecules of DNA. And when everything goes right, the cell divides normally. Unfortunately, that enzyme, when it gets to a thymine dimer, it doesn't know what to do. When it reaches a thymine dimer, the enzyme doesn't know what to put on the new strand of DNA because it doesn't recognize the dimer. So what it will do is it will just place any base there. And often that means an incorrect base. If an incorrect base gets placed in the new DNA, and remember in this diagram, that's the gold color. If a new incorrect bases get placed in this new DNA strand, the next time that cell replicates, that change is going to be permanent. And that's what we call a mutation. What a mutation is a permanent change in the order of the bases in a DNA molecule. So what ultraviolet light can do is create these dimers that then, once the cell goes to divide, are going to lead to mutations occurring. If enough damage is done in this DNA molecule, that cell's not going to survive. If enough damage is done, if enough thymine dimers are created, that cell's not going to be able to survive. But if the damage isn't too bad, before the cell replicates, there are repair mechanisms that can come in and fix the dimers. So in other words, if a microbial cell, a bacterial cell gets exposed to ultraviolet light and gets some dimers in its DNA, there are mechanisms, there are other enzymes that can come in, they'll find those dimers and they'll repair them. And then when the enzyme comes in to copy the DNA, everything's fine, everything's normal. So there are really two outcomes when a cell gets damaged by ultraviolet radiation. It will either get repaired or it will die. All right. So that's it really for bacteria. That damage is either going to, that ultraviolet exposure is either going to damage it, damage the DNA, or kill it outright. Obviously for you and I, as multicellular eukaryotic organisms, we also have a third thing that can happen, right? We can get cancer from the damage to our DNA. So sometimes our damaged skin cells actually go on to become cancerous because of those mutations. But bacteria don't get cancers, microbes don't get cancers. Now let's talk about two of these mechanisms that repair DNA. And you should know that human cells also have repair mechanisms. So our DNA can also be repaired to a point when it's damaged by ultraviolet light. In bacterial cells, there are two primary systems for repair. One of them is called photoreactivation or light repair. And the other one is called excision repair, or what's also known as dark repair. Now, the reason these systems are called light and dark is because one of them can only work in the presence of light, visible light. The other one can occur in the light or in the dark. So photoreactivation, anytime you see this term photo in a word, it means light. So this tells us that this is a light repair system, a light reactivation system. What the visible light will do, that cell that's sitting in the light, what will happen is it will activate the repair enzymes. And we give these enzymes just sort of a family name. We call them photolyases. So this is a whole group of enzymes. And what these enzymes are able to do once they're active is travel down the DNA strand. And anytime they find a dimer, a thymine-thymine dimer, they can split it. They can cut the connection between the two thymine bases and restore the DNA to normal. Now, excision repair is a little different. First of all, it doesn't need light to happen. The enzymes that are used in excision repair don't even have to be activated. They're already active. They're another family of enzymes. They're called endonuclease enzymes. And what these enzymes do when they find a dimer is they just cut it out. They remove it and they replace it. Actually, other enzymes do this. Other enzymes replace those two missing thymines. So the endonuclease goes down the DNA strand, and anytime it finds a dimer, a thymine-thymine dimer, it removes it. So now you've got a hole in the DNA strand. Other enzymes will come along and replace those two missing thymines and return the DNA to its normal anatomy. So this sounds great, right? Wow, ultraviolet radiation damage can be fixed in microbial cells, but only to a point. In other words, neither one of these repair systems is 100%. Some thymine dimers will not be found and will not be repaired. And sometimes these repair systems make mistakes. So sometimes they miss dimers and fail to break them apart, or sometimes they remove dimers incorrectly and cause additional problems. But for the most part, they're very efficient systems and they do a good job repairing minor damage in the cells. Does anybody have any questions about this process, about what the ultraviolet radiation is doing, number one, and about why that's a problem for the cell? Remember, the DNA molecule, those bases are what constitute genes for the microbial cell, right? Those bases are the code that the cell will use to build proteins. So if a sequence of bases, if a length of the DNA molecule that codes for a protein, if it gets damaged with dimers, now that gene is damaged and the cell can't use it to make normal protein. So thymer formation is bad. It's bad for the cell. Thymine dimer formation is bad for the cell. It can interfere with the normal functioning of the DNA. And as we said, it can lead to mutations down the road. And if there's enough damage, that cell's not going to survive. But, again, as human beings, we take advantage of this. We purposefully expose surfaces to ultraviolet light because we're trying to kill cells. We're trying to eliminate any invisible microbial cells that might be on that surface. And it works very well. And it's not terrifically expensive either, ultraviolet exposure to surfaces. So it's very helpful in terms of our ability to control microbes in our environment. All right, let's talk about our experiment. Now, we have a couple of different objectives today. We have our purpose is multifold. First of all, we want to examine the effects of ultraviolet exposure just on the ability of the cell to survive it. So we're going to expose cells that we've grown on an agar plate. We're going to expose it to ultraviolet radiation for 30 seconds and also for 60 seconds. Now, I know that doesn't seem like a long time, but for unprotected bacterial cells, they have no protection against ultraviolet radiation. That's enough sometimes, not only to damage, but also to kill. So we're going to see the effects of these two time periods of exposure on those cells, whether or not they're able to survive it. We're also going to examine whether or not these bacterial cells can repair damage to their DNA. And since we know there are two different repair systems, one of which requires light in order to function, what we'll do is we'll make doubles of each plate. We'll incubate one of the plates in the light, and we'll incubate one of the plates in the dark. In other words, we'll allow the photoreactivation system to work on one plate, and we'll allow the endonuclease excision repair system to work on the other plate. Now let's talk about the organisms that we're going to use for our experiment. There are two bacterial cells that we're going to be using. One of them is called Bacillus Licheniformis. That's quite a name. And the other one is our old friend Escherichia coli, or E. coli. Bacillus Licheniformis is an interesting organism. Like all bacillus, it's capable of forming spores, right? But it's not a pathogen. It lives outside, it lives in the soil, and it's saprophytic. And that term just means it helps break down and degrade dead organisms. So it's one of the life forms that are out in the natural world that help get rid of dead organisms. It helps degrade and decay dead material. Now, it is a spore former because it's a bacillus, so that tells us it's pretty hardy. It can survive what some other bacteria would not be able to survive, so we'll keep that in mind too. E. coli is a gram-negative rod. Oh, I don't know if I mentioned that this bacillus is a gram-positive rod. E. coli is a gram-negative rod, and we know that some strains are pathogenic. Some strains are not pathogenic. We've talked about how E. coli lives inside of the human intestine, for example. It's an important part of our microbiome. But there are some strains that are pathogenic. And E. coli is what we call an enteric coliform. Remember, this is an organism that you find in the intestine that you would also find in fecal material. So here are the two organisms growing on agar plates. These are quadrant streak plates. Remember, the purpose of making a quadrant streak plate is to grow individual colonies, because sometimes we want to examine those colonies for colony morphology. So we can see some individual colonies of E. coli growing here. They're pretty small, I think you'd agree. They're circular. They're sort of a creamish color. That's a pretty common colony morphology, by the way. Now take a look at the bacillus. This one looks very different, doesn't it? We do have individual colonies here, even though you might not know it at first. This is a colony right here. This is a colony right here. This is 1, 2, 3, 4 colonies side by side. So these are big colonies, right, compared to these. They're very big. They almost look like fried eggs. They have a central sort of darker area, circular area. They have a very irregular margin. They're also sort of a cream color, but they look very different from these colonies. They're very irregular looking. So 2 different organisms, and they have 2 different colony morphologies. So what we're going to do is use these organisms again and expose them to ultraviolet radiation and see what happens. So we're going to use nutrient agar plates for our experiment. And we're going to need a control. So we're going to make 4 nutrient agar plates, and we're going to, I'll say we're going to make 4 plates for each of our 2 organisms. And each plate will be divided in half. Half of the plate will get covered so that it will be protected from ultraviolet rays, and the other half will get exposed to the ultraviolet rays. And the reason we need 4 plates is because we're going to both look at 30 seconds of exposure and 60 seconds of exposure. And we're going to incubate those plates both in the dark and in the light. So this is the breakdown here for the 4 plates. 1 plate is going to get 30 seconds exposure incubated in the light. One's going to get 30 seconds incubated in the dark. Then we have 60 seconds in the light and 60 seconds in the dark. 4 plates for each organism. And on each plate, half of the plate is going to be covered so that it doesn't get exposed. We're going to need that control. We're going to need to be able to show that when the cells are protected from the ultraviolet rays, they grow perfectly normally. We need a control for comparison's sake, so we can see the effect of the ultraviolet exposure. All right. So this diagram up top, this just shows very basically, again, how you make a spread plate, because we're going to make spread plates here. And we're not going to try to grow colonies for counting so much as we're going to try to grow a lawn of cells. We want to grow as many cells as we can. Now, the way that you know you'll get a lawn instead of individual colonies is by making sure that your original broth culture is loaded with cells. Remember, we can look at a broth tube that comes out of the incubator, and we can know that cells are growing if the broth is cloudy. So we want to make sure that the broth is as cloudy as we can get it, so we know we're putting tons of cells in the middle of the plate. We'll take a sample of the broth, we'll put it on the plate, and then we'll use one of our hockey sticks, sterile hockey sticks, to spread that broth perfectly all around every inch of that plate. We'll put the plate in the incubator, and hopefully we'll get a nice lawn of cells growing, at least on the control half. So we'll spread the cells out on the plate, and then before we put it in the incubator, we'll do our exposure to ultraviolet. So see what I've done down here. Here's a plate that I put some culture onto, I put some liquid onto, and then I used a hockey stick to spread all the cells around. Then I covered half of the plate with an index card, and I put the plate into this box. This is an ultraviolet box. These are relatively inexpensive tools that we use in the lab. You can see the box is, it's made out of metal. The sides are completely opaque. You can't see through it. This device right here that is plugged into the wall, this is the bulb that will emit ultraviolet light down into the box. There's also this eyepiece here. This is a viewing port. And if you look through here, if you put your face down into this area, you can see what's down in the box while it's going on. And there's a film across the eyepiece here that protects your eyes from the ultraviolet light, because of course, ultraviolet can harm you as well as microbes. So for each one of the plates, after we spread the cells out, we expose them to UV. Then they go into the incubator and we let them grow. All right, let's talk through these results. For 30 seconds of exposure, on the control side of the plate, and again, that's the side that was covered up, we got a nice lawn of growth. See that? All of that area right there that looks kind of yellowy green. That's just an entire growth of cells. They're not colonies growing here. There's this whole huge just collection of cells that we refer to as a lawn of cells. This is the side of the plate that had the index card over it in the UV box. This is the side of the plate that didn't have the index card over it. This plate was exposed for 30 seconds to ultraviolet light, and then it was put in the incubator. So some colonies grew, right? A fair number of them, really. Look at all those colonies. Now, does this look like this? No, right? They're not growing like this, are they? But they're growing. So these cells we know are alive because they are dividing, but they're certainly not dividing as normally as we would see. So the colonies are much smaller than they would be normally. This happens to be the Bacillus Licheniformis plate. Here's the E. coli plate. Again, this side got covered up by the index card. This is a lawn of cells. You can see how it's sort of this yellowy, opaque color. The agar surface is just covered in bacteria here. That's what we expect. This is the side that got exposed to ultraviolet light. Boy, there's hardly any growth on this side, right? I've got one enormous colony right here. There's another one here, another one here, one here. I think there's one down here in the picture. So some cells certainly are on this side and they're dividing, but I don't have nearly as many colonies here as I do on the Licheniformis plate. So let's think about this exposure for a second. How come an index card was enough? How come I could just place a piece of paper over that plate and protect those cells anyway from the ultraviolet? How come a piece of paper was enough? How come I didn't need to, like, I don't know, cover it with some thick layer of material? What do you think? I put a simple paper card over half of that plate, and those cells were obviously completely protected because they grew like crazy, right? They grew into a lawn. What is it about ultraviolet that means that a little index card will stop it? What is it about ultraviolet that we've learned? It's big limitation that allows me to protect those cells with a simple index card. What do you think? Brooke saying reflection of light and penetration, Sydney, penetration. It's the penetration, right? That ultraviolet can't penetrate that card, or it can just barely penetrate that card. It's just not making it to the cells because it can't get through the card. It just doesn't penetrate very well. That's its big limitation. If you can get the cells exposed directly, you can kill cells. You can disinfect. But ultraviolet is not going to penetrate through any materials. It's just not. That's why when you and I go out in the sun, if we wear a tight weave shirt, like one of those surf shirts, we can protect ourselves from ultraviolet, right? Because it can't get through the cloth. It can't get through the cloth. It has very poor penetration. Now, here's another question. How come when I exposed these cells, the side that did get exposed, I took the lid off. I took the lid off here. Gosh, that doesn't seem like good sterile technique. It's really not good sterile technique, right? But I had to do it. I had to do it. Why? Why did I have to take the plastic lid off of that Petri dish when I put that plate into the ultraviolet box? Why couldn't I have just left the lid on? Same reason, right? Same reason. If I had left the lid on the Petri dish, even the side that I wanted to get exposed would not have gotten exposed because the ultraviolet wouldn't have been able to get through the lid. It can't penetrate. So if I want to get those cells exposed, I have to take the lid off. So you put it into the ultraviolet box, you quickly close the ultraviolet box to try to prevent contamination. But yeah, you've got to, because ultraviolet doesn't penetrate. It doesn't penetrate materials. That's an important thing to remember if you're using ultraviolet as a disinfectant. If you're trying to disinfect, let's say, a countertop in let's say, I don't know, a plant that processes meat for sale in the grocery store. So you've got a countertop where meat gets chopped up and put into packages and sold. And let's say at the end of the day, I'm going to turn on an ultraviolet light because I want to disinfect that surface. If I left a whole bunch of stuff on the countertop, it's not going to penetrate. It's not going to clean off. It's not going to disinfect the countertop. If I left a thick layer of, let's say, not to be gross, but meat residue, if I left a big layer of stuff on that countertop, the ultraviolet's not going to be able to work because it can't penetrate. I got to clean off the counter first and then turn the light on and disinfect the counter. Got to remember that. That's the big limitation to ultraviolet light. All right. So here's an important thing that we noted, and we won't even need to mention again, and that's this. It didn't matter whether we grew the cells in the light or in the dark. We got the same results. Let me say that again. It didn't matter if the plates were in the light or in the dark. It didn't matter for either organism or for either time point. The plates looked almost identical, whether they were put into the light or the dark conditions. And what that tells us is that both repair systems were able to function about the same. Didn't matter. Remember, if we put it in the light while we incubated it, we were turning on the photoreactivation system. If we left it in the dark while it was incubating, we were allowing the excision repair system to work. And it turns out that both of those systems worked equally well because we didn't see a difference between the plates that were from the dark conditions and the plates that were from the light conditions. So that's a result we have already. Here's the 60-second plates. Take a look at this. Here's the side where we were covered. Here's the side where we exposed to ultraviolet. This was for 60 seconds. This is a lawn of cells. There are cells growing on every inch of this agar, creating this film, this yellow greenish looking film of cells. Over here, nothing. There are no colonies over here. If you look at the E. coli plate, very similar results. Here's that lawn. On the control side, on the covered side, we got lots and sorry, this is also Lyconiformis. Oh, I'm sorry. Nope. Sorry, I'm moving things around and I'm not moving my labels. This is the E. coli plate. No colonies on the side that got exposed to ultraviolet light for 60 seconds. Plenty of growth on the side that was protected but nothing over here. So how do you read these plates? All the cells for both organisms, all the cells that got exposed to ultraviolet light for 60 seconds died. None of those cells were successfully repaired. The damage was just too great. And again, that's pretty amazing if you think about it. 60 seconds, 1 minute of exposure killed all those microbial cells, all those bacterial cells. It's pretty amazing. Didn't happen at 30 seconds, did it? Some of those cells were repaired and grew. Some of them died, but some of them got repaired and grew into colonies. But after 60 seconds of exposure, they're all dead. So that tells you something, right? That tells you that ultraviolet kills bacteria. It's a good disinfectant, but you got to expose them for at least a minute or more. According to our results, you've got to expose them for a minute or more. And obviously, most of the time that people use ultraviolet lights to disinfect surfaces, they're leaving them on for, not even minutes, but usually hours. So to make sure everything's dead. But it's pretty amazing that it's that powerful against bacterial cells. A short exposure, that 30-second exposure, some of the cells can come back from that. They can be repaired, and then they can grow and divide, make colonies. But even at 30 seconds, a lot of those cells were killed, right? How do I know that? Well, it didn't grow as a lawn after 30 seconds. It grew as colonies, isolated colonies. Where there were no colonies, those cells were killed. They were dead. But some of the cells survived, they got repaired, and they grew into colonies. Does anybody have any questions about what we've looked at or what it means? Any questions about that? We spread cells over that whole plate. And remember, the part of the plate that was covered up, we didn't get colonies growing. We got a smear, like a whole lawn of cells growing every inch of that plate. So that tells us there were a lot of cells that we spread out on that plate. And when we exposed half of that plate to ultraviolet for 30 seconds, a lot of those cells got killed because there's blank areas on the agar in places. There's nothing there. So whatever cells were there are gone. They're dead. But everywhere there's a colony, a cell survived. And it got its DNA repaired. And it was able to go ahead and divide, into a colony overnight. So we have evidence of repair. We have evidence that at 30 seconds, some of those cells are repairable. Some aren't, but some are. But at 60 seconds, it's too much damage. It's just too much damage. Even with the repair systems in place, those cells just can't come back. They can't fix the damage and they can't survive. So let me summarize what we've seen. Oops, here we go. What effect did 30 seconds of ultraviolet exposure have on the Bacillus Lyconiformis cells? Did it kill all the cells? No. No, it didn't. How do I know? Because I saw some colonies. I saw colonies on both plates, the plates that were in the dark and the plates that were in the light. In fact, they looked almost identical. I saw a lot of colonies, didn't I, on that plate? I'll show you a picture of it again in a minute. So was DNA damage repaired in some of these cells? Yeah. both the light system was working and the dark system was working, because again, both plates looked pretty similar. So would I disinfect a stainless steel work surface in my restaurant with 30 seconds of ultraviolet exposure? Would you? Would you feel good turning on your ultraviolet light in your restaurant for 30 seconds over the work surface where the meat was being cut up? Would you feel good that 30 seconds of exposure was enough? What do you think? No, right? No. No, because obviously enough cells survived it that we grew colonies. Yeah, 30 seconds isn't enough. From our results, at least, you would need 60 seconds at least. Yeah, good. So I put this slide together. These are the same pictures, obviously, but just to drive home the point that we had two organisms, Bacillus Licheniformis and E. coli. We made plates for each organism. We made 30-second and 60-second exposure plates. One plate was incubated in the light, one was incubated in the dark, but it didn't matter. We got similar results. Same with the E. coli. One was incubated in the dark, one in the light, but the effects were the same. So we know both repair systems worked. I do want to ask a question though. I want to know, can I compare this plate and this plate? Can I say that, for example, Bacillus Lyconiformis did a better job repairing its DNA than the E. coli did? Obviously, fewer colonies grew on the E. coli plate, right? I got many fewer colonies, lots more colonies on this plate, even though they're pretty small. So can I make that kind of comparison? Can I say, oh, well, Bacillus Lyconiformis must be better at repairing its DNA than E. coli is because more Bacillus cells survived than the E. coli cells after 30 seconds of exposure. What do you think? Brooke is suggesting not better maybe, but maybe a different pathway. We know, Brooke, for bacteria anyway, that these are the two pathways that they use. We know from previous research that they're going to use light repair if they're in the light while they're incubating, and they're going to use excision repair if they're in the dark. So it's not so much that the pathways are different, they're both using the same pathways. So the question is, how come the Bacillus plate, after 30 seconds, we had more colonies on it than the E. coli plate? Can I determine that? Can I compare those plates? What do you think? It's a hard question, right? It sure looks like it, right? It looks like more of the E. coli died because I got fewer colonies on the E. coli plate than I did on the Bacillus plate. And if you think about it, think about where these two organisms live. Go ahead, question?
Oh, never mind. I was gonna say, maybe E. coli like doesn't stay like too long. That like maybe like shelf life for the bacteria to stay alive than the other, but it does make sense.
Oh, I see what you're saying. We don't have any evidence for that happening. That's a good guess, though. We don't have any evidence that that's what happens in the laboratory with those two organisms. But that's certainly a good guess. But think for a minute about where these two organisms live. Bacillus Lyconiformis lives out in the world, in the soil. And E. coli lives deep in your intestine. Who gets exposed to more sunlight ordinarily? Who gets exposed to ultraviolet radiation on a daily basis. The bacillus does, right? Bacillus Lyconiformis, if you think about it, might have evolved to be better at repairing its DNA because it has to, because it lives in the sunlight. It lives outside in the soil, so it's going to get exposed more. So maybe through evolution, it developed a better repair system than the E. coli did. E. coli just doesn't get hit with ultraviolet light because it lives in the intestine. That's a reasonable speculation, right? But here's the thing. We can't make that comparison in this experiment. This is about experimental design. All right? This is about experimental design. If we wanted to figure out whether Bacillus or E. coli was able to survive better after ultraviolet exposure, 30 seconds of exposure, we would have to make sure that we put the same number of cells on both plates. Does that make sense? You would have to ensure that you started off with the same number of cells. And then you could say whether or not more or less cells survived because you started with the same number. We have no way of knowing how many cells exactly were on that Bacillus plate and how many were on the E. coli plate. It may be that the E. coli plate just had fewer cells on it than the Bacillus plate had. So sure, fewer colonies grew because there were fewer cells to begin with. You see what I'm saying? When you design an experiment and you want to make comparisons between two organisms, you've got to make sure that the conditions are the same for both the organisms. And we didn't do that in this experiment. Ours was what we would call a qualitative experiment. We weren't trying to quantify how many cells survived. We just wanted to know if cells survived. And we saw cells surviving on both the Bacillus plate and the E. coli plate because colonies grew. But you can't make a quantitative comparison there. You can't compare numbers because you didn't put the exact same conditions on both plates. You didn't put the same number of cells to start with on both plates. It's a lot to think about when you're designing these experiments because you have to think about what you're trying to prove or what you want to learn from your results. All right, let's jump back in here. So let's think about the 60-second plates now. And remember, for the 60-second plates, nothing grew. It didn't matter if we were in the light or the dark for either organism. These are the Bacillus Lyconiformis plates. These are the E. coli plates, light and dark. We got nothing. No cells grew on either, or I should say on any of these plates. The control cells grew fine. The side that was covered up, those cells grew fine, but the exposed cells didn't. So what effect did 60 seconds of exposure have? Let's start with Bacillus. Did it kill all the cells? Yes. Was DNA damage repaired in some of the cells? No, at least not successfully, not to the point that would have saved the life of the cell. Some repair might have gone on. In fact, it's likely that it went on. The plates that went into the light for incubation, the photoreactivation system would have turned on and it would have started breaking those dimers apart, but it obviously wasn't enough to save the cell because no cells got repaired to the point where they could grow into colonies. And the same was true for the E. coli. It's likely that those repair systems were working for E. coli, both the light and the dark repair, but it wasn't successful. to keep the cells alive. We know that because no colonies grew. If those repair systems had worked and restored those cells to health, we would have gotten a colony, and we didn't get anything. 60 seconds and it was all killed. So of course, from our results, and of course this was a very simple experiment, we would say, yeah, 60 seconds is enough. 60 seconds is a good amount of time to disinfect a stainless steel surface, although you might want to go even longer than that just to make sure. Just to make sure. So these bacteria, they're very vulnerable to ultraviolet light, to ultraviolet radiation. And the reason they're vulnerable is because they're so small. They're only maybe 1 or 2 micrometers in diameter. Some of the biggest rods that we've looked at, they may be 4 micrometers in length. That's a 1/1000th of a millimeter. And we know that ultraviolet can penetrate up to 2 millimeters. So ultraviolet goes right through, all the way through bacterial cells. They are very vulnerable. They're single-celled organisms. They only have 1 cell. And if the DNA in that 1 cell gets damaged badly enough, that cell's not going to make it. So that's good news for us who are trying to stop bacteria from growing in certain environments. Bad news for the bacteria. You just have to know how to use the ultraviolet light. First of all, you have to remember that it's only useful on hard, solid surfaces. Again, because it doesn't penetrate. Number 2, as I said a minute ago, we want to make sure that we clean that surface before we expose it to the light, the ultraviolet light, because it can't penetrate through a lot of gunk on the surface and get down to the table or whatever solid surface you're trying to disinfect. I talk in lecture about how there have been a lot of attempts in the commercial world to market ultraviolet devices to people, especially during this pandemic. A lot of commercial catalogs and companies have been trying to sell people ultraviolet devices, telling them this is a good way to protect yourself from microbes. And as I said in the lecture, it's buyer beware, isn't it? Because even though they'll tell you, for example, that you can disinfect your mattress with an ultraviolet wand, you and I know that that's just not possible, because the ultraviolet light can only go 2 millimeters down into that mattress. So you can't disinfect your bedding with an ultraviolet wand. You can't do it. It's just not going to work. You can certainly disinfect your kitchen counter or maybe your bathroom counter, some nice solid hard surface, but you're not going to disinfect your bedding with ultraviolet. It can't get in. It cannot penetrate deeply enough to do that job for you. Yeah, so it's definitely buyer beware. And of course, nowadays, I also mentioned in lecture, we have these wonderful devices, the big canister devices that have ultraviolet bulbs all the way around them. And you can clean, for example, a room in a hospital, clean it real well. And then you can roll this big machine into the room and turn it on and shut the door. And that ultraviolet light is going to go off in all different directions. It's going to hit a lot of the surfaces in the hospital patient room that sometimes we miss with a sponge or a mop. So ultraviolet, it has very good application in healthcare for disinfecting solid surfaces. And it's not particularly complicated. You just have to be sure not to expose your workers to ultraviolet light without protection anyway. They need safety goggles on if they're going to be in ultraviolet light, and they need their skin to be protected from it, because we will get our DNA damaged too. That's exactly right. All right, any other questions? You should be able to complete your lab homework now. You should be all set for that. I will see you again on Monday during the lab period. And that's all I have for you today. So if there are no questions, I'm going to let you go to enjoy this beautiful afternoon. Hopefully you'll have a chance to get outside. It's really lovely. All right,
Wait.
everybody.
I have a question.
Yes.
I think I may have missed it because I did go to the bathroom earlier, but I did ask about number 4 in the quiz. What happened?
Number 4.
Yeah.
You know what quiz?
It's the most recent quiz.
Oh,
It was
that's the one I repaired. I fixed. Yeah. That was a question that Blackboard graded incorrectly.
yeah.
And that's my fault, because when you make a quiz in Blackboard, what
Yeah.
you do is you write out all the possible answers that you want to
Yeah.
give to the students. And then I, as the instructor, have to check off which answer is correct. And if I don't do that, Blackboard chooses the first one as the correct one. So the
Oh.
correct answer was just in the quiz incorrectly. I had to go back in and I had to say, no, this is the correct one. And then it fixes the quizzes after that. Yeah.
So my quiz is fixed.
Yes.
Okay. Thank you.
You're very welcome. Yep.
Sorry about that.
Oh,
I
no
know you
problem.
probably explained it earlier, but I probably
No
wasn't
problem at all.
laughing. Thank you.
All right, everybody. I'm going to let you go. Enjoy the rest of the day.
Thank you.